from single digit figure to multi-digit, then all over United States of America, then Africa, finally all over the world. This year is the 35th year after we got the HIV in this world that was started from Los Angeles, United States of America. In 1982, the Center for Disease Control scientists, they named it for this deadliest disease as acquired immunodeficiency syndrome, that's AIDS. In 1983, when Dr. Francois Barros-Sinosi was examining the biopsies received from lymph node biopsies received from HIV patients from United States of America to Pasteur Institute, France, he was able to found some abnormal particles in the lymph node lymphocytes. Next one, please. So this is the first HIV picture taken by Dr. Francois in Pasteur Institute. So these particles are viral particles. So that time they did not know whether this is a virus or something else. They call it as abnormal particles. So this particle is attached with the cell membrane. I think this is CD4 receptor. So cell membrane of lymphocytes of lymph node tissues. This was the first picture taken by Dr. Francois Barros-Sinosi in Pasteur Institute. This was the first picture of HIV to this world. Next one, please. She is Dr. Francois Barros-Sinosi. She is the first lady found the HIV. Next one, please. He is Dr. Lou Mandakner. He was the superior to Dr. Francois Barros-Sinosi. Dr. Lou Mandakner and Dr. Francois, they isolated the HIV and characterized it. And simultaneously, he is Dr. Robert Gallo. He was the US scientist working in the Maryland University as well as National Institute of Health, USA. Simultaneously, he also started isolating and characterization of the virus. These three people, they isolated the HIV and characterized it. In 1984, they found it. This is the retrovirus group, and this belongs to lentiviral characters. They named it as human immunodeficiency virus, HIV. In 1984 only, we got the name HIV. Next one. Please stay in the previous slide. So for this wonderful work, in 2008, these three people were awarded Nobel Prize for discovery of HIV. Next one, please. Why there is no cure for HIV? This is also a $1 million question. Since there is no cure for HIV, there are 33 million people are suffering in this planet, and 37 million people already passed away from this planet due to this deadliest disease. The HIV gets integrated into our host chromosome. The HIV integrated cells play as reservoirs. That means it constantly produces the viruses. That is called the reservoir. No drug is able to eliminate the integrated HIV or to destroy the reservoir cells. No vaccine is successful so far because the virus is keep on getting mutated. It doesn't have highly conserved region. Next one, please. So the integrated DNA gets integrated into our host chromosome. That is called either integrated HIV DNA or integrated DNA or proviral DNA. This is the proviral DNA integrated into our host chromosome, we have 46 chromosomes, 23 pairs. These integrated HIV having 9 genes. This 9 gene possessing HIV genome gets integrated any one of our host chromosome. Mainly at the time of integration, whichever the chromosome or gene having the highly functional work, highly busy genome, it will sense that the HIV genome sense that chromosome and gets integrated into that. From that integration time, then next moment it will start producing transcription. It produces the HIV viral RNA, then translation. It produces the viral proteins. The protease enzyme. This helps to assemble the viral protein to become fully grown viral particles. So the viral particles budding out of the cell by bursting the cell membrane, and this budded immature virions becomes matured viral particle ready to infect other cell like this. So here, this kind of matured viral particle here and coming here. You imagine this viral particle coming here. So this viral particle has receptors called GP120. This GP120 gets attached with the CD4, and this receptor is called GP41. This GP41 comes here and attached with the CCR5 or CXCR4. This CCR5 and CXCR4 will be, either one will be present in the human host, depends on their genetic makeup. Gets sent after attaching with the receptor, gets entered into the here, gets entered into cell by piercing the cell membrane and uncoating and release the RNA here. This RNA is being transcribed as cDNA by reverse transcriptation. After the reverse transcriptation, there is a complex called proviral integration complex. This proviral integration complex have the power and affinity to go and integrate into our host chromosome. So here, for this purpose here, for the integration purpose, here one enzyme which is produced by the HIV disease called integrase enzyme. This integrase enzyme helps to integrate into our host chromosome. That is why no medicine, this is a typical disease in the medical history, in the medical world. This is because the viral DNA, that is cDNA becomes DNA, gets integrated into our gene. Whenever our gene gets transcribed, these 9 genes also starts transcription. This is because when once the HIV is integrated into our cell, whenever our body starts functioning, this HIV also starts producing. So no drug is able to go pierce the nuclear membrane and go and act here. This is called pharmacogenomics. The pharmacogenomics not yet developed to go here and act and do the function, so on. Next one, please. So, when we talk about what is the existing treatment, so what treatment we are giving to the HIV patient, what it does. The existing treatment, they call it as HART, hyperactive antiretroviral therapy. In medical field, they shortly call it as ART, antiretroviral therapy. So which are all the antiretroviral therapy? In beginning, they were given single drugs like reverse transcriptase inhibitors, protein inhibitors, fusion blockers, and the integrase inhibitors. Nowadays, through operational research, they molded it should be given as combination of therapy. That is CRT, combination of antiretroviral therapy. But either single drug or combination of drug or whatever may be, this is not able to eliminate the integrated DNA, and it is not able to destroy the HIV reservoir. Next one, please. So what is the action of ART? There are 4 drugs I said in the previous slide. The first one is fusion blockers. The HIV gets entry through mainly CD4 cell membrane, because whichever the cell has the CD4 receptor, that cells are called the CD4. So gets entry through the cell membrane. The fusion blockers block the entry of the HIV here and arrest the entry of the virus. Then next one is reverse transcriptase inhibitor. So I said previously, after entry it releases the RNA, then RNA becomes cDNA, cDNA gets integrated into our cellular DNA. So these reverse transcriptase inhibitors inhibit this reverse transcriptase enzyme not to produce any cDNA and to integrate into the cellular DNA. So next one is integrase inhibitors. These integrase inhibitors inhibit the integrase enzyme and arrest the integration of HIV cDNA into the cellular DNA. The fourth one is protease inhibitor. So after doing all the things, I already told, from integrated DNA, the virus RNA is being transcribed and protein assembly, then budding of virus and mature virus and so on. So this protease helps to budding of virus. So when we arrest the protease with the help of protease inhibitors, the budding of virus is being stopped. So the mature virus will not come out from the cell and go and infect the further another cell, next cell and further cell, another cell, whatever it may be. So, but these 4 kind of ART doesn't have any effect of already integrated DNA, because this DNA is already integrated. No one can stop preventing it to produce the RNA transcription and protein assembly, but it will be go on circulating into the cytoplasm of the cell. Once the cell is getting broken, these RNAs come out and wandering into the plasma. And these drugs is not able to destroy the whole cell also, either breaking it or whatever may be. So if we want to cure the HIV permanently, either we have to eliminate this already integrated DNA, or we have to destroy this whole cell. If we destroy this whole cell, the whole integrated DNA and cellular contents, cellular viral contents will be diminished and vanished and washed out from the circulatory system. This is the strategy I developed. Next one, please. We have reverse transcriptase inhibitors, NV inhibitors, protease inhibitors, but even then we cannot cure the HIV. Why? Because here the integrated DNA is constantly producing the HIV, embedding into the cell. So if you want to cure the HIV, either we have to destroy the cell as a whole, or we have to eliminate this 9-gene HIV integrated DNA from our 46 chromosome. Unless we do this, we cannot eliminate the HIV or cancer like these genomic integrated diseases. As far as I know, only 2 diseases. One is HIV and cancer. Instead of this integrated DNA, HIV DNA, cancer, there will be oncogene. So this concept can be applied to cancer also. Next one please. So what is my work? So when I talk about my work, I have unearthed, excavated and explored our Tamil traditional medicine for potential activities towards elimination of integrated DNA and to destroy the HIV reservoir cells as a whole. First I started with the herbal drugs, the potential selected herbs which can do something in the immune system and destroy the virus as a whole. That means whichever the herbs is able to replenish the immune cells. When we talk about the immune cells have the natural killer cells, macrophages, cytotoxic T lymphocytes, some kind of, this kind of cells is able to destroy the another cell. This strategy we want to cure the HIV. So here our aim is to destroy the HIV harboring cell by stimulating the cells like natural killer cells, macrophages and cytotoxic T lymphocytes. Among these killer cells, the CTL, cytotoxic T lymphocytes, particularly having one peculiar character. It doesn't allow any foreign protein harboring cells, any foreign nucleotide incorporated cells. It will sense that particular cells and it will go and destroy that. I used this strategy through herbal medicine. I selected the herbs, whichever the cells, whichever the herb is able to generate continuously to these immune cells through maybe 10 years of 1 decade of hard work. Then finally I could be able to achieve some encouraging result I obtained directly from the HIV patient, even though I did a lot of works in vitro animals. So my surrogate marker is the live HIV patient, whether he is getting improved than ART or not. This was the question all the way. I don't mind about the ethics, all the things, because I'm not doing the crime. I'm only giving the benefit to the people. This is the deal between me and my patient. And after getting the success in the herbal drugs, I added metals and minerals due to some strategy, because I have filed it for patent. I cannot say all the science, I cannot disclose all the science what I got in my work. So sorry, once again I ask sorry for that, not to revealing all the scientific things here. Then I prepared the crude drug, I given it to the HIV patient. So after giving the crude drug, I observed the patients were getting better improvement than the herbal drug. Next one please. Based on action of my crude drug and the observations I made I got it from the HIV patients, those who are taking crude drug. I proposed one theory that is called Prakash theory of metal drugs. Prakash theory of metal drug. Based on my theory, I engineered the metal as in nanoscale and attached the biomolecule which has been derived from my previous herbs as ligands. These molecules are called Prakash. Next one please. Now I have produced 4 individual Prakash molecules. This is the complex molecules having metal atoms and biological molecules. This is new to this world. There is no, as far as I know, there is no metal drug which have been approved for the treatment of diseases orally. There is 2 metal drugs, silver sulfadiazine and some other chromium drugs that are all only for external use. There is no drug approved for the internal use for the particular disease or so on. So I have produced so far 4 molecules After the patent, if I release it, anyone can produce metal drugs based on this theory, which will be benefited to the world than the existing drugs. So, if anyone wants to do the research after the publication of my work, it will be easy if you follow the theory, then that time you can remember me. So this theory is useful for my research. As far as HIV patent is concerned, I have filed 2 patents in HIV cure. No one in the world in HIV cure filing the patent, either in India or US or international filing. As Dr. Krishnan said, GDP is important for the country to release the fund towards the research. So my GDP is only my individual GDP. So far, nearly, I should not tell the figure, maybe some income tax officers are, this is the public forum. Last 22 years, I have spent my own income only to produce these Prakashing molecules. So this is the nightmare for the Western people. If you say about the metal, they will say, this is the toxic. So this is the Prakashing drugs, it is being given to the HIV patient. They are very, very happy with this. I will tell you about this later. So regarding the patent, so I have the patriotism. Our country, India, should gain this name. Even though a lot of my US friends, they are keep on calling me to settle in the US. I didn't want. So I have to succeed from this soil, even though there is no government support here. So that's why I have filed the patent. This is the beginning for the HIV community, not for me. This is the betterment for the HIV community. Since I have worked 22 years in this research, I have dedicated my life to the HIV community. One patent, I was talking about the patent. One patent in HIV cure is HIV with crude drug, and another one with the synthesized molecules. Why synthesized molecules? This is the science. When we talk about the crude drug, it will be having thousands of molecules. It will act better, but as far as science is concerned, the scientific community will not accept it. I learned it from my experience. The international community people, they did not accept, even though I produced the cure. They said it's a crude drug, there is no science. Then only I got borrowing and I got unearthing the science from the crude drug. That's how the Prakashing born. Next one, please. Effects of Prakashing complexes towards HIV cure. So PCs are able to stimulate the immune cells continuously. That means continuous proliferation of the immune cells. These molecules act somewhere in the outer membrane of the cells and some sort of signal transduction mechanism. It induces the new kind of signaling and stop producing the immune cells. As well as it produces the cytokines. As well as species renew the stem cells. Once the stem cells are being renewed, it will produce all kind of progenitor cells, RBCs, WBCs, some other organ cells, whatever it may be, along with the biochemical molecules, that is the cytokines. Once these cytokines, renewal of stem cell and immune cells are being produced continuously, we can live more than 100 years. So that is the natural fighting mechanism against the infectious disease. The immune cells will take care of the infectious disease, I mean antigens, which are all coming out, enter into the host. The immune cells will take care of it. And the cytokines, it will take care of the non-infectious diseases. It will regulate all the enzymes and hormones, like that. So as far as side effect is concerned of the Prakashings, so can you imagine when this chemical got the capacity to renew the cells? Will it do the damage to the cells? Anyone can say? One agent is able to produce the cells like food. What we are taking the food? For the cell growth. Do you imagine the food will damage the cells? So I have engineered this molecule not to produce even a single side effect as nutrients, as dietary supplement. If I take 10 capsules now, you no need to call the ambulance. This is another kind of ART drug. It is being given to the HIV patient all over the world, recognized by WHO. If I take 5, definitely you should call the ambulance. Next one, please. So, as far as treatment of the HIV patient with the PC is concerned, the patient has to take this species for about 36 months. During the treatment with the PCs, there will be a controversial surrogate clinical parameters we will observe that is opposite to the ART mechanism. Apart from that is controversial blood parameters we will notice, that is increase of viral load and decrease of CD4, whereas in the ART, once we start the ART to the HIV patient, the CD4 will increase, HIV will decrease. Whereas in the Prakashins, the viral load will increase and CD4 will decrease. You may ask the question, your drug is wrong. I will explain you why in the next slide. So this viral load increase and CD4 decrease, this kind of fluctuation of the immunological marker and virological marker will be in the first year only. In second year onwards, the CD4 keep on increasing like ART, the viral load become 0, and finally the HIV DNA is being transcriptionally inactivated. That is called the functional cure. In some cases, the viral DNA and the reservoir is destroyed by the CTL, stimulated by the PCs. They will get permanent cure. This is what happening. For this treatment, people are coming from USA, France, all developed countries, from all developed countries to Navakas. Next one, please. I told, in case of Prakashin treatment in first year, the viral load will be elevated and CD4 will be decreased, whereas in ART, the CD4 will be increased and viral load will be decreased. Let me first talk about the ART control mechanism. When we talk about ART, that is reverse transcriptase inhibitors, NV inhibitors, protease inhibitors. So the reverse transcriptase inhibitor inhibits the reverse transcription and stop the multiplication of HIV here. So the coming out mature virus is prevented by NV inhibitors, and protease inhibitor also stop the replication. So in blood level, the number of virus will be reduced and the CD4 cells level will be increased. Since this ART is able to arrest the HIV replication, this CD4 cell is not destroyed. That's why the destruction rate of CD4 cells and pass out rate of HIV virus is reduced. That's why once start the ART treatment, the CD4 is keep on increasing and the virus is keep on decreasing. Whereas the PCs is able to destroy the whole reservoir. Imagine this cytoplasm contains many viral particle, maybe one cell contains 2000 virions like that inside. Once we break the cell wall, where these 2000 virions will go? It will go to the plasma only. So if we break one CD4 cell, we will get 2000 extra virus. So we are reading the viral load. That is different mechanism. We are saying only the HIV RNA. We are not seeing the whole virion. If we see the whole virion, if we have the technology to count the whole virus, it will be reduced. Here, lot of double standard HIV RNA will be there. All will come to plasma. We have the technology, reverse transcriptase PCR, quantitative reverse transcriptase PCR, only to count the copies of the HIV RNA, not to count the whole virion. That's why we will get increased level of HIV RNA copies in quantitative PCR. So, coming to the logic, if we break this CD4 cell, we will get extra 2000 HIV RNA copies. When we give the feces to the HIV patient, how this cell, this reservoir is being broken and being destroyed? Because the feces is able to produce more immune cells. In that immune cells, there are lots of CTL is generated. That CTL, cytotoxic T lymphocytes, sends this virus harboring cell and destroy it whole. Once CTL destroy it, there won't be an environment for this integrated DNA to keep alive. So that is the logic. Once we break the reservoir, all the HIV RNAs will come out and there won't be an environment to be alive of this integrated DNA. Then there won't be a further transcription and there won't be a further production of the virus. If we keep on destroying these HIV harboring cells, one fine morning in the HIV patient, there won't be a virus DNA, HIV integrated DNA harboring cells. That is the logic behind. So, how the CD4 is getting increased? If we keep on destroying the CD4 by the CTL, this number will reduce. The HIV RNA copies come out to the plasma. We are enumerating the HIV copies as viral load in the plasma only. So, because of increasing HIV RNA copies due to the destruction of the reservoir in first year, the HIV viral load will be elevated, and because of destruction of the whole reservoir, this is the CD4 cell. We are enumerating the CD4 cell, and the CD4 cell number will be reduced. After destroying all these reservoirs during first year of treatment with the PC, there won't be any virus harboring cells. There won't be any HIV integrated DNA to produce further HIV. So from the second year onwards, the viral load will be decreased and CD4 will be increased because there is no transcriptionally active HIV DNA to produce further virus to infect further cells. That is the logic I handle. Next one, please. So I told about the two kind of treatments for HIV people. The one is existing treatment that is WHO recognized, that is being given to HIV patients all over the world. So this is the ART, what the WHO HIV prevention program is followed all over the world. In our government also National AIDS Control Program, in that also they are giving the ART. They say this has to be taken throughout the life. But as far as mine is concerned, this is the third HIV. There is two HIV. HIV1, HIV2 and HIV. This is third HIV. I see in my experience all over the world, no man alive without any side effect after 10 years. Last week, one gentleman came from France. He was, with his consent only, I am telling his personal things here. I said, I am going to talk in the conference. Can I share your experience with the conference, because this is a public forum, it is unethics. He given the consent, okay, you can talk about me. With his consent only I am telling. I am following the Helsinki Declaration, ethics in my treatment, I think so. So he told, I was affected 20 years, 22 years, I am taking 10 years of treatment. What treatment? This ART. Why you came here? I heard in the France newspaper being published, you are on Indian, prepared some medicines through your normal medicine from your research. So this is no side effect. I want that. I want to avoid this ART. I asked him, what side effect you are facing? He said, my vessels are getting burst, he told. Bursting of the vessels. What kind of bursting? So in the right and left, so vessel got blocked and burst. Still many more vessels in my body, it has to be operated. I am doing one by one. I am a nurse in France hospital, he told. So of course, I explained, you are taking ART, it will elevate all kind of lipids, especially bad lipids. It will give the way to aggregation of platelets in your blood vessels, wherever it may be there, maybe in heart, maybe in brain, and it will clog the blood flow. You may have to operate it. You have the facilities in France, you are getting operated, but here so many people are dying without doing that. So this is the example for the ART, how much side effect is being given. So when we talk about Prakash, I said if I take 10 capsules at a time, no need to call the ambulance. So the people, those who have taken this medicine for 3 years, this is unapproved drug only. I am fighting for the approval. So how I am giving the treatment to the patient? This is the deal between me and the patient. I am doing the good thing for the patient, whoever seeks to my treatment. The France man is coming all over the way by spending flight ticket and everything, finding my address and asking appointment, requesting for give my treatment. Should I give or not? Should I say this is unapproved? I will not give. Which is correct? Which is the humanitarian action? Should I follow the law? Or should I do the help to the people, those who are seeking me? This is unanswered question. I am a human being. Whoever comes to me, I am giving that. At the same time, I am fighting for the approval. As a scientist, as a good things present in this Prakash, I want to give this medicine as many as possible. For this purpose, I have approached National AIDS Control Organization and the Union Health Minister, Union Health Secretary for the approval and for the further financial support to develop it further. Unfortunately, I did not get any green signal from them. Next one, please. So, since this Prakash molecule is new to the world, new metal drug to the world, there is no side effects. It is able to modify our immune system by stimulating the cytokines, by proliferating, manipulating many more field, several types of immune cells and renewal of stem cells. I am doing this for HIV. Anyone can do the research for cancer, diabetic and so on, immune related, hormone related, enzyme related diseases. I hope this Prakashin will work for many diseases. If anyone would like to do the research in these Prakashins, whether I get a patent or it is under pending stage or after the patent, I am ready to give the material. Whoever wants to conduct the research in that. If whoever wants to do the research before I get the patent, I will give this material with prior non-disclosure agreement. Next one, please. So, I would like to thank for my friend Dr. Naraj Srinivasan for giving me this opportunity.